A-Level Biology 9700, Paper 4, Variant 2, October, November 2024, question number 8. Figure 8.1 is a diagram of a motor neuron. Neuron is the functional unit of nervous system. So neurons are of three different types: sensory neurons, relay neurons, and motor neurons. They have given us the motor neuron. There are small differences between the structure of the sensory neuron and motor neuron and relay neuron. Motor neuron is just like the motor neuron is larger in size as comparison to relay neuron. Relay neuron is just like the motor neuron, but there is small difference in the structure of the sensory neuron and motor neuron. The sensory neuron has larger dendrons but smaller axon, whereas the motor neuron has large axon. So this part is called as axon, and this part is called as cell body, which has nucleus inside and cytoplasm is also there. And these small branches arise from the cell body. They are called as dendrons, and dendrons are further divided into dendrites. So D is dendrite. So axon has the cells which are called as Schwann cells. And Y is the cell body. And students, uh, the nerve impulse move uh, from, first of all, the stimulus is there. Stimulus is a change, and the change is always received by the receptors, which can be the sense organs. So receptors from this information is transferred to sensory neurons, which take this information to the central nervous system, and from relay neuron to the motor neuron, and from motor neuron to effectors, and from effectors perform the response, which can be the muscle contraction. So in this question, we will discuss the how muscle contraction takes place. So here you can see the next question is, name a type of cell that forms a synapse with the structure Z. Here you have seen the neuron, motor neuron. So the nerve impulse always move from cell body towards the axon. So it means the information is coming from the relay neuron towards the motor neuron. So we have synapse. What is synapse? Synapse is the gap between two neurons. On one side of the dendrites of the one neuron and axon terminal of the other neuron. So dendrite is on this side. So this is dendrite. It means there will be the axon terminal of the relay neuron. So in this way, so there will be the gap in this way, a small, you can say, relay neuron. So it will be the axon terminal. So it will be synapse. So it will be small distance or the gap between dendrite of the motor neuron and axon terminal of the relay neuron. Axon terminal of relay neuron. Next part of this question is table 8.1 shows some of the events that occur during muscle contraction. They are not listed in the correct order. And those events are A. Calcium ions diffuse out of the sarcoplasmic reticulum. Myosin heads bind to actin. Sarcolemma depolarized. Sarcomere shortens. And calcium ions bind to troponin. T-tubule system membranes depolarized. Binding sites on actin exposed. Myosin heads tilt. Tropomyosin moves and troponin changes shape. So these are the different events which takes place during a muscle contraction, but they are not in sequence. We have to make their sequence. And to make their sequence, first of all, we must know the structure of a muscle fiber. So here I am going to yes, muscle fiber, structure of muscle fiber. Muscle fiber, you can say it is multinucleated. It is long cylindrical structure that run the entire length of the muscle fiber. So I would like to say the muscle fiber is multinucleated. It has many nuclei. So secondly, each muscle fiber is a single elongated cylindrical muscle fiber surrounded by a delicate layer of connective tissues. And then muscle fibers are multinucleated, meaning they have multiple nuclei located at the periphery of the cell. The cell membrane now. Cell membrane cell membrane is called as sarcolemma. And then cytoplasm is called as sarcoplasm. And then sarcolemma has invaginations called transverse tubules. Invagination. Invaginations of sarcolemma. So they are called as T-tubules. And then there is sarcoplasmic reticulum. Sarcoplasmic, just like the endoplasmic reticulum in a germinal cell. So sarcoplasmic reticulum's function is to release the calcium ions, to store and release the calcium ions, which play a crucial role in muscle contraction. And then mitochondria. Many mitochondria are present which produce ATP produce energy in the form of ATP for muscle contraction. And then myofibrils are also there. So I would like to discuss about sarcomere. Which is a region between two Z disks and between two disks. Distance I'm giving a brief so for your reminder only. And there are thin filaments and thick filaments. Thin filaments are called as actin. And thick filaments are called as myosin. So the actin filaments are actin, and they have tropomyosin and troponin. These are the special type of proteins, regulatory proteins. We say that tropomyosin and troponin. So these are the produce interaction between actin and myosin. So these are two types of regulatory proteins. They are associated with actin. And then myosin, they are thick filaments. They are thick filaments and they consist of many myosin molecules. So before going in detail of this sequence, I want to show you a diagram. With the help of this diagram, I will explain the different sequence which will take place here. That is, first of all, what will happen, how do this action potential will start, or the action potential is reached over here. So then I will fill in the blanks which has been given. So first event which takes place is the action potential arrives at the muscle fiber. So muscle fiber, there will be the action potential on the muscle fiber. So what will happen at that time? At that time, sarcolemma will depolarize. Sarcolemma, which is the cell membrane of the muscle fiber, it will be depolarized. And then after the depolarization of sarcolemma, you know, the T-tubule system membranes are, which are the invagulation of sarcolemma, they will also be depolarized because the action potential will spread into the muscle fiber. I would like to write over here in this action potential. So it will depolarize the sarcolemma, which will depolarize to T-tubules. So as a result of this, what will happen? The calcium ions diffuse out of the sarcoplasmic reticulum. You know, the sarcoplasmic reticulum, just like the endoplasmic reticulum, they store and release the calcium ions. So calcium ions will diffuse out from sarcolemma. So after the diffusion of them, calcium ions, those calcium ions. Here you can see this is the myosin fiber, which is thick filament, and thin filament. This is the thin filament. Troponin and tropomyosin is there. So you can see when the calcium ions are released, so these calcium ions will bind to troponin, exposing binding sites for myosin, because here actin and myosin, they will bind together to make a cross bridge. So after the release of calcium ions, you can see they will bind. So these calcium ions which are released over here, they will bind to troponin and then exposing the binding sites for myosin when they will join. So these binding sites, so these are the binding sites, these which has been shown with dark blue color. So at that time, what will happen after this binding? You can see the bound myosin rotates its head, producing a power stroke. You can see in this way. So it will move its heads, and it will bind to the troponin. the troponin, it will occur with calcium ion bind to troponin, and then after this binding, what will have, the ADP is released, and there will be the ATP will be used for this purpose, and ATP molecules bind to the myosin head. So they will bind with the myosin head, and then actin and myosin detach. First of all, so this is the situation which we say that is the formation of cross bridge. So when they are joined together, you can see this is the situation where the cross bridge is formed. After this, the ATP molecule bind to the myosin head. It will bind, so it will provide it energy. So this energy will help to for power stroke. And after this, the troponin uncovers will get separate from, detach from each other, and then ATP is hydrolyzed. So in this way, this whole sequence of events will be continued. So I'm going to write all this again. As after the release of calcium ions, so calcium ions will binds with troponin, and it has been shown over here in this picture. And then it will uncovers the binding site, binding sites, and then myosin will bind. So here you can see the myosin binds, and then after the binding of myosin, cross bridge, formation of cross bridge. So this is a cross bridge. And then this myosin head will tilt. Will tilt, and it will detach the actin and myosin from each other. So detachment, they will be detached from one another. So this is the result of the sliding filament mechanism, that is the sarcomere shortens. So sarcomere shortens. So after this explanation, I'm going to the question where we have to fill up the table as shown. I have explained. Now I will write down. First of all, we see sarcolemma on the action potential, sarcolemma depolarized. After the depolarization, the T-tubules are also depolarized. After their depolarization, the calcium ions diffuse out of the sarcoplasmic reticulum, that is A, and then there will be the calcium ions will bind to the troponin. Already they have given us J, and J is the troponin changes its shape when calcium ions bind to the troponin. And then after the J, there is the sequence I when the binding sites are uncovered on the actin. So this is I, and then the binding sites on the actin will expose, so this allow myosin to bind. So the binding of the myosin will be there, and after this, the myosin heads bind to actin, so this will form cross bridge formation will takes place over here. And after the formation of cross bridge, the myosin head will tilt. When it will tilt, this is the power stroke that slides the actin filament. So it will be H. Again, they has given us T, and D is when sarcomere shortens. So this is the result of the sliding filament mechanism. So in this way, the event of sequence in a muscle contraction will take place. Now, last part of this question. Lambert-Eaton myasthenic syndrome is a rare disorder of the neuromuscular junction. A person with LAMS produces antibodies that bind to the voltage-gated calcium channels on the presynaptic node. One symptom of LAMS is weaker muscle contraction. Suggest and explain why LAMS lead to weaker muscle contraction. Well, students, Lambert-Eaton myasthenic syndrome is characterized by the body's immune system mistakenly producing antibodies, which that attack voltage. These antibodies attack to voltage gated calcium ions, calcium channels. Uh, so located on the presynaptic nerve terminal. So these calcium channels are crucial for the release of acetylcholine, a neurotransmitter that signals muscle contraction. So, when students, when a nerve impulse action potential reaches the presynaptic terminal, it causes these voltage gated calcium channels to open. So the calcium opening of these channels allow calcium ions to flow into the presynaptic terminal. So the influx of calcium ions triggers the infusion of synaptic vesicles containing acetylcholine with the presynaptic membrane. This fusion leads to the release of acetylcholine into the synaptic cleft. So then acetylcholine then diffuses across the synaptic cleft and binds to acetylcholine receptors on the postsynaptic membrane. This binding initiates a series of events that ultimately leads to muscle fiber contraction. I repeat all these events. And students, the acetylcholine is a neurotransmitter which is transferred. So these, the calcium ion channels, presynaptic muscles, and the release of acetylcholine and neurotransmitter is on the when a nerve impulse arrives. So what happens with then? It reaches to the presynaptic terminal. So it causes these voltage gated calcium channels to open. Calcium channels open, and the opening of these channels allow calcium ions to flow into the calcium ions flow into presynaptic terminal. So this is the presynaptic terminal, presynaptic membrane, and this is the postsynaptic. So acetylcholine it will down acetylcholine, it will move here over this presynaptic membrane or presynaptic terminals. So it has to move towards the other part, the postsynaptic. So this acetylcholine is released when it fuses with the presynaptic terminal into this over, and there are calcium ion channels, and then this acetylcholine presynaptic. So the influx of calcium ions triggers the fusion triggers the fusion of synaptic vesicles. So after this fusion leads to the release of acetylcholine into the synaptic cleft, and then acetylcholine then diffuses across the synaptic cleft and bind to acetylcholine receptors on the postsynaptic muscle membrane. So this acetylcholine, which will be released over here, it will fuse over here in this, and then acetylcholine receptors on the postsynaptic membrane. So this binding initiates a series of events that ultimately leads to the muscle fiber contraction. So in this way, the acetylcholine is released into the synaptic cleft, and from synaptic cleft into the post synaptic membrane. So in this way, the muscle contraction process takes place. Now, what happens when the process LEMS is there? The antibodies block or reduce the function of these voltage gated calcium ions. So the result in what happens when there is LEM? Reduction calcium ions influx. It means there will be the less calcium enter the presynaptic terminals in response to a nerve impulse. Less calcium ions will enter the presynaptic terminal, and then less release of acetylcholine into the synaptic cleft, as I have shown you over here. So less binding acetylcholine on the muscle membranes into the synaptic cleft, and due to which the reduced, you can say, reduced acetylcholine or reduced amount of acetylcholine bindings to its receptors on the muscle membrane leads to a weaker signal for muscle contraction. So, therefore, LAMS leads to weaker muscle contraction because the antibodies interfere with the crucial steps of calcium dependent acetylcholine release at the neuromuscular junction. So, you know, the antibodies attack on voltage uh, gated gates for calcium. So, what happens after that? So the due to less reduction of calcium ions in flux, there will be the less release of acetylcholine, and due to less release of acetylcholine, so less binding of acetylcholine on the synaptic cleft will take place, and the weaker signal for muscle contraction will be produced. So in this way, LAMS affect the muscle contraction, or it leads to the weaker muscle contraction. So it was all about question number 8. Thanks for watching. Please like, share, and subscribe my channel for more questions on papers.